Hi, my name is Jason Deng from HITS platform, WuXi AppTec. So the HITS platform provides products and services to cover a wide range of capabilities to support our clients' small molecule early discovery. So for today's talk, I'm going to focus on the recent development of discovering RNA targeting small molecules using the Deng technology. So RNA targeting small molecules is a new modality of therapeutics. So this is exciting, and especially for people like me who come from the protein small molecule discovery world. But initially, we also got puzzled by the question that for all the drug design experiences and the tools that we've built over the years for protein small molecule discovery, will it still be applicable for RNA? So fortunately, it comes to light that there are some key concepts that still apply. So first and foremost is that RNA is like protein, so its biological function depends on structures. So it is true that if one can design a small molecule ligand that can specifically interact with the RNA target and can subsequently change or block its structure, such ligand may be able to modulate its biological functions. And second, that some of the RNA possess complex and deep pockets. Such pockets are critical for a ligand to have specific molecular interactions for its target selectivity. Not all of those RNA have such deep pockets, and thus the presence and the quality of such pockets can be a useful metric for assessing the chemical drug abilities of a given RNA target. And third is that the concept of drug likeness can still be a useful guiding principle for us to design RNA small molecule drugs. So it is there's evidence that showing small molecule ligands that are drug-like, which are in the green box region there, they tend to bind to complex tertiary and secondary structures, whereas ligands that are not drug-like, they tend to bind to simple secondary structures, such as secondary structures with repeated elements. So this gives us hope that we may be able to use some of the existing tools from the protein small molecule drug discovery world for RNA ligand discoveries. So this including the HTS, Dell screenings, fragment screenings, as well as structure-based and virtual screening methods. We believe that Dell technology has its own advantages for identifying RNA small molecule ligands, because by nature, Dell is a affinity-based method, and it is relatively straightforward to set up the screening conditions. Also, generally, it has faster turnaround time for the assay result. So here's just a brief sort of introduction to the Dell. Basically, Dell is a library of chemical structures within which each of the chemical compounds is physically attached to a DNA barcode. And because of the nature of this compound, we can very quickly and sort of synthesize a large size library. And then we come to the affinity selection. Once we have the Dell libraries, all we need is purified protein or RNA target that can be immobilized to affinity matrix. And in our case, we've been using magnetic beads. So we started affinity selection by incubating the target with our Dell libraries. After the selection step, we can identify the binders where we can go through synthesis and the validation steps. So, but for the RNA specifically, there are some conceived challenges. So the first is that RNA structures are fairly sensitive to experimental conditions. So we want to find a robust and rapid method to verify the RNA structure integrity and binding pocket before the selections. And then the other well-perceived challenge is that the interaction between the DNA tags and the RNA molecules during the selection steps. So we wanted to develop a method that can mitigate such risks. Right. So for the RNA structure verification, traditionally people using crystallography to determine the RNA structures. Here is an example of tetrahydrofolate rival switch and its crystal structures, along with its folate ligand and its putative binding sites determined by the crystal structures. Getting those structure information can be time-consuming and labor-intensive. So we actually went to evaluate an alternative method for fast structure folding analysis, which is so-called a shape map. It's a method published in 2015 by a group of researchers in University of North Carolina. So in this method, briefly, we use electrophile reagent that can selectively isolate the two hydroxyl groups on the open region of the RNA molecules, such as on the open loop or on the bulge section. Such isolation will introduce mutations during the prime extension and the cDNA prep steps. And we can read into the mutation profiles that would give us information to determine the secondary structures of the RNA molecules. So taking the THF rival switch as an example, we have it without the binding ligand. So we observe that there's a shift of the mutation profiles upon the binding to the THF ligand. And we also saw that some of the sites that actually corresponded, some of the changes along the RNA actually corresponding to the ligand binding sites determined by crystal structures. Then with that information, we also predict the folding of the RNA molecules under our selection conditions. It is actually overall consistent with the ground truth from the crystal structures. And also we can use some of the sort of frequency of the mutation to profile the ligand binding site, which is also consistent with what's been shown in the crystal structures. So this actually gives us a method that we can use to verify or QC the RNA structures for our selections. Right. So next is actually we want to address the issue about the DNA-RNA interactions. So a quick anatomy of WuXi's DNA tag that basically consists of both a double-strand DNA region and a single-strand DNA region, which within that single-strand DNA region, we have a unique molecular identifiers consist of random chimer sequences. So in one of the initial experiments, we actually saw we incubate our Deng molecules with RNA. We saw abnormally high level of binding over its protein counterparts and non-target controls. And after some sequencing analysis, we actually detected some motif-specific binding of our single-strand DNA to the RNA molecules, and the sequence actually aligned to that top open loop region on our target. So actually here we actually employed a method called DNA zipper So basically, we use a polymerase that convert the single-strand DNA region to double-strand DNA, and that actually blocked that single-strand motif. In our experiment, we show that after zipper treatment, we effectively remove all the motif binding on that single-strand region. Right. So just showing on the plot here, right? The blue lines are without the zipper. With the zipper treatment, the detection is basically very baseline. Actually, in a separate experiment looking at RNA G4, in this case, actually, we have a natural ligand, this quinoline. So we actually add a DNA tag to it and use it as RNA2 compound to spike into our DL selection. With this RNA2 compound, we're able to track the behaviors of those compounds during the selection process and understand how the selection performed. And in our observation, we saw that this DNA tag 2 compound showed a significant binding. Shows significant binding to our RNA target, whereas the negative control ligand showed a baseline binding. But this only happened with the zipper treatment. Without the zipper treatment, our negative ligand also showed substantial binding to our RNA target. And this background binding is RNA dependent, as we in our no target control, we don't see that background. So the zipper treatment was able to remove a significant portion of the RNA-dependent binding noise. So we sent the samples to NGS and just to confirm, the zipper treatment also, indeed gave us a much higher sort of binding signal of the 2 compound, also with a minimum level of background noise due to the DNA interaction. Right. So with that method, with those 2 methods, we just want to we actually performed 2 pilot DL selections against these 2 targets, THF ribose switch and RNA G4. So the selection itself is fairly straightforward. We set up the RNA G4 as our main target, and we use this G4 mutant as our non-structured RNA control together with NTC as sort of our negative conditions. So after selection, we were able to identify some substituted iso-indolent structures that seems to be fairly selective to our RNA target. And similarly, in the other target, THF ribose switch, we also found some interesting linear scaffold that also shows selectively bound to the THF target. And this is just another example showing in another library, we can identify different scaffolds as well. So this actually comes to the summary of my presentation. So we demonstrated that DL can be adopted for RNA ligand discovery. We use shape map method for fast RNA structure verification. And we observed strong single-strand RNA, single-strand DNA and RNA interactions in our tested RNA targets. And with this zipper method, we can, after we convert the single-strand DNA to double-strand DNA, we can effectively remove this background. So all the data here is unpublished, so we're still in the stage of validating those RNA hits with biophysics and functional assays. And we are also actively expanding our case studies to cover mRNA and microRNA. And also because there is such a powerful tool, we're also trying to investigate the specificity and the selectivity of the RNA pocket. So this is the So actually, all the work is done by our team in Shanghai. It's a dedicated professional group of scientists working very hard to expand the capability of Dell, and all the credit goes to them for this work. And I left my email here just for any questions related to this work or any information or find out about our services from the HIS department. Please reach out to us. Thank you.
All right. Thank you very much.